So test cross, okay, we actually want to know, okay, particularly why we perform a test cross, because, for example, if I have the yellow seed versus the green seed, green seeds, you definitely can pinpoint out what is the genotype. It must be small y, because they are homologous, homozygous recessive. Correct. Okay. But the thing here is yellow seed, it can be either big Y, it can be big Y, small y. So which one? Then in order to find out this, then we can perform a cross called test cross. So the idea of test cross, actually, we want to determine the genotype of an individual, okay, a genotype of individual, whether they exhibit the dominant trait is because of the homozygosity or heterozygous for that particular traits. Like for example, yellow seed, big Y, or big Y, small y, then we can perform a test cross. So this is why we need to know why we need to carry out test cross. And how we carry out test cross. Basically, a test cross, we always to take cross the organism, show the dominant phenotype, and in order for these dominant phenotypes to be passed on and shown in the offspring, we need to cross them with the homozygous recessive. Are you clear? For this dominant phenotype to be passed on, right? So it means that this dominant phenotype, we don't want them to be masked, then definitely we need to cross it with the homozygous recessive. Okay? Why? Because the effect of the effects of the recessive allele always will be masked by the presence of the dominant allele. Okay, so therefore phenotype of any offspring will reflect the genotypes of unknown parents. So the known parents, it must be homozygous recessive. So we studied with the monohybrid test cross. So we use back the example that we already learned previously. So it means that you will be more familiar with this example. So we do know that the seed color determined by two allele, big Y and small y. Big Y code for yellow seed, small y code for green seed. Okay, so it means that if we have the yellow seed, it definitely either big Y, or big Y, small y. Green, you don't need to figure out because we always, it always will be homozygous recessive. Okay, so in order to find out the genotype of yellow seed, whether they are homozygous dominant or heterozygous, we cross them with the homozygous recessive green seeds of the garden tea plants. So based on the outcome, okay, based on the outcome, you will be able to know. Okay, so how we base on the outcome? Look at it. So if, okay, I use if is a homozygous dominant, you cross it with the homozygous recessive, then you can see that because all gametes 100% will be big Y. So this one 100% will be small y. So this one's unknown. Okay, it can be either big Y, or big Y, small y. So if there's a big Y, then you can see that if I cross like this, if I carry out a test cross, then definitely all will give me big Y, small y. Correct? So means that this one all will show me yellow color seed. So if all, the results show me all yellow color seed, then I know the tested subjects here, must be homozygous dominant. But if let's say it's a heterozygous, when it cross with this, then you can see that half of them big Y, half of them small y. So when you cross it with all small y, so then I will have two outcomes. I may have the yellow seed, I may have the green seed in the ratio of 50-50 or 1-to-1 in this case. Can you see that? Are you clear? So it means that if the offspring show me 1-to-1, then I know that this one must come from the heterozygous. Okay? So a quick one, okay? Let us look at this. For monohybrid test cross, if my F2 garden pea plant show me all yellow color, so means that yellow, it must have one big Y, correct? Right? And then no green color, so means that another one also must by the big Y. Okay, if I have yellow seed and green seed, so yellow seed, big Y, green seed, small y, correct or not? So means that it must have one small y, this must have one big Y. So means that the F1 phenotype must be big Y, small y, or heterozygous. Okay, must be heterozygous, correct or not? Okay, get the idea? Clear? So now we continue with dihybrid test cross. Similar. In dihybrid test cross, okay, we're going to talk about two genes, okay? So one is for yellow seed, green seed, another one is round and wrinkled seed. So if I have yellow round seed, so basically it can be homozygous dominant for both, or one heterozygous, okay, or both heterozygous. Are you clear? So what we're going to do here, we have to cross it, because we want to cross it with the, you want to test cross, you always test cross it with the homozygous recessive green wrinkled seed. Okay, homozygous recessive green wrinkled seed. Okay, so look at the result. If I get all yellow round seed, all yellow round, so what I know here, yellow round, look at this, yellow round, it must have one big Y, one big R. Correct. One big Y, one big R. Right? And then because we test cross it with the homozygous recessive, so this one comes from the homozygous recessive. So big Y earlier come from who? It must be big Y, big Y, big R, big R. Can I see that? So the green color here is contributed by who? By the green wrinkled. Okay, this is how we find out. If I have yellow round seed and green round, okay, we have a yellow round, so big Y, big R. Green round, so it must be small y, big R. So from here, I know that big Y, small y, big R, big R. Can you see that? Can you find out? Okay, very simple, right? So yellow round seed, yellow must be big Y. Round must be big R, must have one big R. Yellow wrinkled, so means that one big Y. And then wrinkled must be small r. Can I see that? So big Y, big Y. Big R, small r. Okay, yellow round must have one big Y, round. Can I see that? Yellow wrinkled, so big Y, wrinkled must have small r. Okay, green must have small y, round, big R. Green, small y, small r. So means that a mixture of big Y, small y, big R, small r. Can I see that? So this is how we can use the test cross based on the result of the offspring, figure out what is the dominant trait. So what is the dominant trait? Sorry, what is the genotype of the dominant trait? Okay, so with this, we have done for the test cross already. Okay, quite simple. This is something like logical thinking. From here, we will be able to figure out what is the genotypes of the dominant trait. So with this, I've done for today. Okay, all the syllabus that